Today we're honored to be speaking with Dr. Yosef Shiloh. He is the David and Inez Myers Professor in Cancer Research at the Sackler School of Medicine at Tel Aviv University. He has received the 51st Annual AACR GHA Clues Memorial Award, and he's also just won the Jerusalem Prize. First of all, congratulations.
Thank you. Thank you very much.
Would you give us a little background on the rare genomic instability syndrome called ataxia-telangiectasia?
It's a rare genetic disorder, a very severe one. In fact, it's a multi-system disease. It affects the nervous system, leading to severe cerebellar atrophy, which then consequently leads to progressive ataxia, lack of balance and a lack of coordination of the fine movements. And in addition, it affects the immune system, causing severe immune deficiency with recurrent infections in some patients. There is chromosomal instability in this disease and cancer predisposition. And in addition, very severe, very acute radiation sensitivity. So ironically, the patients who get cancer and are treated with radiotherapy suffer from severe radiation reaction, which limits very much the treatment of cancer when it occurs in these patients.
How did you become involved in AT research?
I met a family with patients many years ago. It was in the summer of 1977, and I was looking for a subject for my Ph.D. thesis. I met a family with AT, and the striking clinical picture of the disease made me decide on the spot to do my thesis on AT, and I've been investigating AT ever since.
Do you remember the specific moment in which you realized you had discovered the AT gene?
Yes, actually, I remember it very, very well, very vividly. I was teaching on that day, and I just came back from my class, and one of my students was showing me a result she got just a couple of hours earlier, and we were examining various genes that we isolated from the region to which the gene, the elusive gene at that time, had been mapped. And she was analyzing the structure of one of the genes that we had isolated from that chromosomal region in patients from a specific family that lives in Israel. And it was clear from that result that the gene was, in fact, part of it was deleted in the patients. That is a very serious, very severe mutation that inactivates the gene completely because part of the gene is simply missing. So it was clear to us that we probably had the right gene in hand.
What was it like to be able to identify and clone the AT gene and ultimately provide for the first time a diagnosis for those who had gone without one?
It was a very exciting moment because I had been working on AT already for close to 20 years. And that this was actually a very long and tedious effort that lasted for 8 years in the lab, during which the entire lab was focused only on identifying the AT gene. In addition, it was, as it always happens, a close race with other labs to identify this gene, which had a very high public profile, so to speak, and therefore attracted a lot of investigators who tried to identify it because it was clear that the gene has a very important function just based on the clinical picture of AT. And of course, it involved cancer predisposition, which is, of course, already had been a very hot topic of research. So we were relieved to see that we have the right gene, and in fact, further experiments in the lab indicated that it was only one single gene responsible for this disease, as opposed to what was the common knowledge or common assumption in the field until we identified the gene, that there were several different genes responsible for this disease in different families. So in fact, the word single, which appears in the title of the cloning paper, is very important. And it meant that based on this gene, diagnosis can be done very accurately, diagnosis of the disease, carrier detection, and prenatal diagnosis in every family with AT around the world. So that was very good news for the families. But obviously, that was just the end of the beginning, because then we had to embark on a new mission, understanding the gene's function and specifically the function of its protein product. So we switched gears completely from molecular genetics to protein chemistry, and since then we've been studying the ATM protein.
Your work has significantly increased our understanding of DNA damage response and repair. What are the clinical and translational implications of this research related to cancer?
Well, during the last few decades, it became clear that cancer is caused by changes, alterations in the cellular genome. And therefore, maintenance of genomic stability is essential for cancer prevention. So understanding the very complex mechanisms that guard and maintain the stability and the integrity of our genome is at the heart of cancer research. So it's very intimately related to understanding cancer causation. Now, on the other hand, the radiation and some of the drugs that are used for cancer therapy are themselves DNA damaging agents. And therefore, understanding the DNA damage response, which is the major axis that maintains the stability of our genome, is also essential for improving and refining treatment modalities for cancer. So that's why understanding the mechanisms that maintain genomic stability are such a hot field now in cancer research. And I guess it's not a coincidence that two of the awards that were given at this meeting, the Princess Takamatsu Lectureship and the Clues Award, were both given to investigators of these mechanisms.
Well, thank you so much for joining us.
Thank you.